My name is Leslie Smith. I'm Emeritus Professor of Computing at the University of Stirling. And before I start this presentation, I want to give a very quick description of how I got, of my journey into neuroinformatics, how I got interested in it. I started off working on parallel processing, and then in the mid-1980s moved into neural networks, and gradually moved towards working on neuroscience, cognitive neuroscience, and then neuroscience itself. And I became interested in the analysis of neuroscience data. And I had a project working with a couple of people in different universities in Scotland. And our part of the project was about producing tools that could do data analysis. So we did this, but then unfortunately, the groups that we were working with weren't managing to get their cultured neurons to stay alive for very long. Indeed, not for long enough for us to be able to get any sensible data to analyze. So that sent me out to look for electrophysiological data that our group could analyze. Well, that proved quite difficult. We did eventually get some data from Georgia Tech, from the Pine Potter group there. But the difficulties that we had there and the issues that arose there led us to become interested in sharing of data. Then my research fellow, Namon Metetwa, and I went to a meeting at Newcastle University in the north of England, and we gave a talk there with one slide about sharing data, and it really rang a bell with the people down there at the Institute of Neuroscience. And from that, we set up the Carmen project, which I'll talk about some more, and that got me deeply involved in neuroinformatics. Okay, so now I'm going to actually run the presentation. Here we are. So I'm going to talk about the effective sharing of neuroscience datasets and what the problems are in trying to achieve this. And that's where I'm from. A very beautiful place when it's not raining. So I've said a bit about how I got involved in data sharing in neuroscience and the range of data in neuroscience and why. But now I want to say about why sharing is important but difficult. And that really is to do with the robustness of experimental work, allowing it to be repeated, allowing people to do the same experiments or the same analysis again. Then I'm going to talk about the Carmen project and about sharing electrical measurements, electrophysiological measurements, talk about what it aimed to do, what it managed to do, and what was learned. And then I'm going to talk about international collaboration, the International Neuroinformatics Coordinating Forum, Neuroscience Without Borders, Neuroscience Information Framework, the Human Brain Project, and eBrains, and then finish off by saying a bit about where we are now and some old and new problems. Now, neuroscience data comes in many, many formats because the brain exists at all these different levels simultaneously, all the way from molecules all the way up to the complete nervous system. And people who work on these areas work and create data on these different aspects of the neural system, all the way from molecular neuroscience, synaptic neuroscience, neurons and microcircuits and circuits and maps and systems, and the whole system altogether. And by working on all of these things, they create different amounts of data. Of course, one's interested in bringing all these bits of data, all these areas of data together, but that's a big job, an interesting job. People are interested in sharing data at all these different levels. And data sharing for neuroscience is particularly important. Now, physicists, chemists, astronomers have been sharing data for decades. But data sharing matters, particularly in neuroscience, because when you're looking at neural systems, no two neural systems are the same. No two brains are the same. And even the same brain looked at today and then tomorrow is going to be different. So even more so than in physics and astronomy, the need to be able to repeat experiments is really high. In addition to this, the experimental data that people get is always different. It's always full of some noise. And the way in which you analyze that data may change the exact results that you get. So you want to be able to take the same raw data and analyze it in different ways. And then in addition to that, you want to do validation across a number of different experiments. So neuroscience is hard. That's why I've got a picture. You might wonder why I have a picture of two oak trees here. And the reason for that is that these oak trees are both recognizably oak trees, but they're very different from each other. And the brains are like that as well. If you wanted to understand something about oak trees rather than about one specific oak tree, you'd want to do experiments on a number of different trees. And that's where we are with data sharing. You want to be able to do the experiments and the analysis on a number of different areas, on different brains, on different sets of data from different brains. So neuroscience data, as I said, consists of a large variety of different kinds of data, all the way from atlases, where you're looking at global brain structure, right the way down to connections between neurons, the connectome, right the way down to the micro and nanostructure of neurons themselves, how the membrane is, vesicles, vesicle release, ion channel structure and operation. And then you become interested, in addition, not just to that, but also in neuropharmacology, the way in which neurochemicals alter the way in which the brain works, and electrical and ionic movements, which seem to be the way in which neurons communicate with each other, or at least one of the ways in which neurons communicate with each other. So why am I talking about sharing now? Because neuroscience has been around a long time. And the idea of sharing data goes back at least 15 years and probably really a whole lot more than that. The International Neuroinformatics Coordinating Forum was established eventually in 2005, and that's a major international organization. And then even before that, Dan Gardner and Gordon Shepherd's Neuroscience Database Gateway started in 2004, and that then led into the Neuroscience Information Framework. And there are a lot of databases. There's a list of them, a long list of them on Wikipedia. In some areas of neuroscience, sharing is more advanced than in others. Neuroimaging in particular has got a particularly good way of sharing data. But many of the other areas are not so good at that. And the Neuroscience Information Framework has a whole database of databases, which I'll talk about later. So in neuroimaging, you end up with a 4-level system. That 4-level system is similar to 4-level systems, or these level systems used in sharing other kinds of data. At the very bottom, you've got semantic web technologies, followed by specification techniques and a core vocabulary. That really amounts to some kind of an ontology. And ontologies are critical for enabling people to share data. Above that ontological and vocabularic level, you have the actual experimental data, the workflows and the results. And that's what the users would normally work with. So neuroimaging data is shared quite effectively using these kinds of techniques. And this is the NIF dataset types. And as you can see, there are many different kinds of data in there, all the way from services, service resources, all the way down to different kinds of sharing. This particular one is organized by resource type and website status and so forth. So NIF gives you access, or at least a map, of the different kinds of neuroscience data sets. So what's the problem? Why are we still talking about this? Well, it's not a solved problem. Vast amounts of data of many different types are created by large numbers of laboratories, and there are large numbers of databases for these different areas. But they're not all integrated with each other. And sharing data introduces requirements of various sorts. At the very bottom level, at the ontological level, you've got data formats. You need to be able to have data in a format that you can share. You need to know about the format in order that you can interrogate it and get the data out of it. At the metadata level for the data, you need to have descriptions of the data that are useful, that allow people to find the data that you want and to be able to use the data in an appropriate way. And then there's metadata for the context of the data itself. What was the animal doing? What sort of neurochemicals were there in the culture? How was the brain slice being perfused. And integrating the data across all these databases entails all of the above so that people know how to be able to use the data. Here in this talk, I'm going to concentrate on electrophysiology because that's what I know most about. So if we look at electrophysiology, electrical measurements, what kinds of measurements are there? You can have minimally invasive measurements where you're putting, for example, an EEG cap on somebody, which is a large number of different electrodes. That can go on humans or indeed on other animals. And then you have tomographic data, PET scans and MRI scans, generally on humans, but also on other animals. And these are non-invasive. You can work with the brain as it is. Invasive techniques are like electrophysiology, where you're actually putting electrodes into the brain, electrocorticography, where you're putting electrodes on the surface of an exposed brain, or electrode-based electrophysiology with single or multiple electrodes, or possibly with patch clamp techniques, where you're actually recording with a clamp around part of the neuron itself. And then in addition to that, there are imaging-based techniques, optical electrophysiology, often using transfected rhodopsin. And what you're looking at there are changes in light given out by these transfected cells in response to changes in the voltage there. And then there are culture-based systems, either with neuronal cultures grown directly on top of electrode arrays, such as the one shown here from multi-channel systems, or brain slices, where a part of the brain is extracted and kept alive by perfusing it. So these are the different kinds of the data sources in electrical measurements, and they all generate large volumes of data. So the Carmen project that I alluded to right at the beginning of this talk was a project that really aimed to allow people to share neuroelectrical measurements, electrophysiological and others, in an effective way. Code analysis, repository, and modeling for neuroscience. And thanks to the late Colin Ingram for that acronym. We were very ambitious. We aimed really to produce an e-laboratory for electrophysiology, to allow people to work directly over the whole of electrophysiology using a remote database system like this. I'm going to talk about the project itself, say who we were, what we built, what we achieved, what we didn't manage, and what we learned. It was a UK project funded by the Research Councils with 11 different UK universities, ranging in the north from St Andrews and Stirling down to the south of Plymouth, including Imperial, York, and other universities, with collaborators elsewhere as well, including Edinburgh, Berkeley, Washington, Tokyo, St Louis, Aberdeen, Seoul, New York, Pennsylvania, Boston, and Brazil. So a large number of people were involved. There's a picture of us from, I think, 2005, 2006, down in Newcastle. That's one of our first meetings. One very sad thing, though, was that one of the people who really was pushing this project in the first place was Professor Colin Ingram, the co-head of Newcastle University's Institute of Neuroscience, who died extremely suddenly in December 2013. We had funding from 2006 to 10 from the UK Engineering and Physical Sciences Research Council, and then follow-on funding from 2010 to 2015. And Colin Ingram and I and others were in the middle of writing further proposals when he unfortunately died very suddenly. We ran out of time and money, and our writing of new proposals was seriously damaged by Colin's very sudden and very unexpected death. We had a portal, portal.carmen.org.uk. It's not there anymore, but it was there for a fairly large number of years. So what we were trying to do, we were looking at the previously existing neuroscience research process, where people would collect the data from their experiments, process the data, analyze the data, publish the data paper, and then dispute and discuss it. And then the referees would come back and you'd change the paper. And then the data from that work would be held on to by the researchers in case they wanted to write another paper, and maybe held on for a bit longer, and then the PhD students would graduate and the research fellows would move on to lectureships, and the data would stay on the computer, and then the computers would be replaced, and the data might be kept on some CDs somewhere, and they would eventually be lost. What we wanted to do was to replace that by having a virtual secure environment where once you'd done the experiment, you could keep the data and share the data, processing the data and analyzing the data on that experimental system, so that once you published the paper, the data would still be there. You could refine the technique, you might apply new experimental techniques, new analysis techniques, you might apply them and work with new experimental data as well, and do comparisons and so forth. But the idea was that the data would remain available and remain able to be used by yourself and perhaps by others as well, indeed. The overall common architecture looked like this. On the right-hand side, we had the data, raw and derived data, derived data being data that's been processed, structured metadata, data about the data itself, and that's critical because you'll never be able to find what you're looking for unless you have good descriptions of that data. And in addition to that, good, accurate descriptions of those data are absolutely critical in order to be able to reuse the data in an appropriate and correct fashion. And then there were the services themselves, the analysis and model code that allowed you to do things with that data. Moving leftwards from there was a compute cluster. And then on the left-hand side, you have the actual portal. You have a web portal. In addition, we had rich clients that allowed you to be able to process the data from this directly without necessarily using a web browser. Of course, the intention was you would use the web browser most of the time, but remember, back in 2006 to 10, web browsers were not very good at doing graphical display of data or anything like that. In addition to that, we had a security system. Now, the security system was absolutely critical because people want to share data, but initially they maybe don't want to share the data, or they want to share the data just with their colleagues and nobody else. Or maybe they're working with another group somewhere, like so the Newcastle group was working with groups in California, and they wanted to share data with the California group. Then you want to be able to do things with that data, services. But of course, you want to do sets of services, not just single services. You want to be able to maybe low-pass filter the data, followed by spike detect the data and spike sort the data. In addition to that, you need a workflow to do that. In addition to that, there was a registry which gave the capability through the web of searching through the metadata and the analysis code for the data you wanted and the analysis techniques that you were looking for. So we created a service-based system with workflows run on a machine in the machine room in York University, accessed through a browser, also accessible over the internet through other visualization tools, but still using internet-based protocols. The idea was that the experimenter would upload the data and the metadata to the server, and there it would be processed by services or by graphically composed sequence of services, otherwise known as workflows. And in addition, when you did this processing, you would also update the metadata so that you were recording not just the data, but what had happened to the data, not just the results of your analysis, but the particular details of the analysis that you had used. That was all with a view towards reproducibility and recreatability. Now, there were lots of cult issues, and they weren't all technical. I'll come to the technical issues in a minute. Not everybody wanted to share their data sets. Some people were very keen to keep just holding on to it. It was their data. Creating detailed electrophysiological neuroscience data is quite difficult. And some people become very attached to their data, although it has to be said that the funding agencies generally are very keen indeed that data should be shared. But that's happened more and more in the last 10 or 15 years. And then there's the issue of when one might expect to share a data set. At creation, immediately, once you've published the paper, once the researchers have completely exploited the data set and published a number of papers about it, or perhaps never. And we had researchers, neuroscientists, who had different neuroscientists had different views, and each of these views was certainly espoused by some neuroscientists. And then who should one share it with? Nobody at all? Well, that's unusual. Normally research takes place in groups of people. So you want to share it with your immediate collaborators, or maybe with some other people that you might be wanting to work with, or with the whole research community. We wanted to be able to support all of these possibilities, except perhaps never sharing a data set. Well, we did support that too. And we created a fairly sophisticated security subsystem in order to allow all of the above. And then, of course, there are issues that are more technical. Raw electrophysiological data comes from lots of different types of sources, and different manufacturers often have their own proprietary formats. In addition to that, some researchers make up their own data sets and make up their own equipment, and they have their own data formats. And they're all different from each other. There was some assistance with data conversion. The NeuroShare DLLs had been put together and allowed people to interrogate data from different proprietary sources. But keeping the NeuroShare up to date was difficult because the proprietary manufacturers change their data formats, or not so much change them as extend them as their equipment changes. And of course, in addition to that, all the metadata needs to be recorded as well. The data source, the electrodes, how many are there? Where are they? What's their geometry? The amplifiers, what kind of amplification is taking place? Filtering, are we low pass filtering, high pass filtering, band pass filtering? And what kind of filter are we using? And what are the needs on that filter? And then there's information about the animal, the brain of the animal, about the location within the brain if it's a live animal, or the location of where the brain slice was taken from. In addition, you need contextual information about the stimuli being provided to the animal, the temperature, what kind of preparation you're using, and that's particularly the case for brain slices and for cultures. Carmen Project produced a document about this on the Nature Proceedings. We produced a document called MINI, the Minimum Information About a Neuroscience Investigation, with a view to saying to people how much data they had to have in order, how much metadata they had to have in order to be able to make this useful. That was one of the things, one of the first publications from Carmen. So handling a large variety of incoming data types was necessary. Handling derived data was important as well. And data needed to be used by the users for graphical display, or by services, analysis software, or workflows, chains of services. And of course, in addition, in a workflow, the output of one service was likely to be the input to at least one other service. So it was critical that we had these services able to use all the different kinds of data from all the different manufacturers for different data formats. And that implied some degree of data translation. We had to be able to have resources access a standard data format, or at least access something that looked like a standard data format to it. We needed an environment where data could be interpreted both by human users and by machines and by the workflows to know whether the data being supplied was the appropriate kind of data. In addition, you wanted to avoid data having to be moved about too much. You're talking about large volumes of data. And you want the services to be able to verify that the data is a valid input type and to use that data in an appropriate fashion. So there you're interested in things like, for example, what kind of numerical representation is being used for the values? What's the sampling rate at the lowest level? And then you need to be sure that the data types you're looking at are appropriate. If I want to do spike sorting, or if I want to do spike detection, then I want to do that on a data set that's been high-pass filtered. If I want to do spike sorting, then I probably want to do that on a data set that consists of spike times and extracts of the raw data from that time. So we need to have the right kind of data going in. So the workflow system really needs to be quite sophisticated. It has to be able to interrogate and understand the data, whether remote or local. And the workflow editor has to be able to get the right information to verify that the service it's about to run is appropriate. In addition to that, you want to be able to get just parts of the data. You may have a 2-hour-long set of recordings from a 64-element array or a larger array than that. But actually, what you want to process is the data set from some small number of electrodes in the 10 seconds after some particular stimulus. So you want to be able to get an extract of this data and just apply your processes, your workflows, just to that part of the data. And then, of course, nothing stays the same. As experimental systems change, new data formats, new data types are created. And you can't really specify these in advance. So we needed to be able to have a system that could cope with what was coming along in the future, create services that would accept new data types, or at least change data types, do this in a consistent manner so that it was backward compatible, and being able to verify that things were appropriate. So how does one solve these kinds of problems? First of all, we have to have some kind of generic metadata system that allows people to essentially fill up a form describing exactly what their data is like. So when you upload a data set, you have to be able to provide the metadata as well. So in addition to uploading the data, you need some kind of tool to allow the uploading of the metadata as well. And indeed, you want that to be a sophisticated tool, because asking neuroscientists to upload the metadata for each and every data file is unlikely to work. They won't do it. So we'd like to have some way of automatically uploading the metadata, and that needs a data standard. And the data standard that we tried to put together was something we called neural data format. And this was a major task that we had to put together in order to allow not just the services to use it, but the users to be able to upload data and get it converted into this kind of format internally at Carmen. It consisted of a number of basic data types: continuous time series, events, other event data like stimuli, segments, which is sections of time series data, which you use if you want to have a particular extract from a time series, generic matrix data, we try to be forward-looking, and image data as well. And the content's described using XML, which means that you can add new things in, allowing the API, Applications Programming Interface, to be backward compatible. So the NDF consisted of the data and the metadata with an XML configuration file. And in addition, you could put vendor data files into it. It allowed different numerical data types, from 8-bit integer to double precision. It allowed downloading of data regions of interest, partial data access. It allowed workflows to be able to tell whether something was appropriate. It supported images and image sequence data, and supported multiple data files for each child. It was a major task, and we had a user interface that allowed people to do things with this. For example, here is the system that allowed people to choose a particular data channel in a particular time. The API, the lowest level one, was written in C because it had to be efficient. Remember, you're dealing with large volumes of data. And this was written in C. I don't want to go into the details of that. Probably more importantly for a lot of people was the MATLAB toolbox implemented on top of that C API. And that allowed full protection, autocorrection for misused data types, and this was used inside Carmen for Carmen service code processing. There was also a Python toolbox as well, which was used to allow Python services to be able to use NDF. And this whole lot was implemented at York primarily by Bojian Lang, and of course others at York too. Then we had workflows. Workflows are interesting you need to be able to do the same kinds of things to lots of different data sets. And a workflow consists of a set of services, a set of things that you do one after the other after the other. Low-pass filtering, spike detection, spike sorting. High-pass filtering, high-pass filtering, spike detection, spike sorting, and then maybe display as one particular example. And you might want to analyze bursts as well. Now, there's lots of different high-pass filters that you might use. There's lots of different spike detection techniques that you might use. There's lots of different spike sorting techniques that you might use. And you might want to analyze the same data sets in different ways to see whether the results are similar to each other. So workflows are useful, and you want to store them, and you want to be able to put them together in a way that's appropriate, and we provided a graphical interface for that. And in addition to that, the system would check that the data type coming into a service was something that that service could actually accept. So that's Carmen. Who used it? Primarily, it was used by internationally distributed research groups. People wanted to share the data that they had and the techniques that they had for analyzing that data with others from elsewhere, with other groups or other people in their own research groups elsewhere. It was a very effective way of sharing data and sharing some processing tools. Some data was made publicly available, and many services were made publicly available, but not as much as we'd hoped. We really hoped that people would analyze their data, do their work on the data, and then make their data publicly available so that the general computational neuroscience community, for example, could use those data sets inside their systems. But it didn't happen as much as we'd like. There were technical problems. Of course there were technical problems. We were dealing with large volume data movements, multiple gigabytes. That was difficult and slow. It's still difficult and slow, but it was even more difficult and slower 10, 15 years ago. Quite a lot of the existing software didn't work well under stress. A lot of it was being taken from research-based software, and when you put it into a production environment, it needed a lot of work to make it work properly. There were multiple browsers. Some people used Netscape. Remember Netscape? Some people used Internet Explorer of various levels. And providing an effective user interface meant using technologies that weren't really up to it. Way back in 2006 to 2010, JavaScript was not as well standardized as it is now, though that's still a problem. In addition, when we were writing the software, we used a lot of Java in the client interface. But later that became unpopular because people were concerned about security on it. And we had an interesting problem with the servers. They began to show their age. We bought a lot of the equipment back in mid-2006, 7, 8, 9, 10. By 2015, it was old equipment, but we didn't have the money for equipment replacement. And at that time, cloud systems were not really developed enough. Certainly at the beginning of our work, they weren't developed enough for us to be able to use them. And there was other software problems as well, of course. Security was good. We provided a very sophisticated security system, but it was difficult enough for people to use. It wasn't that easy to use. And it wasn't proven to be good enough for neurologists to use it for actual patient data sets. We'd have had to prove our security to a greater extent than we did. Supporting multiple services was difficult. They were written in multiple languages. We supported Python and we supported MATLAB, but some things were written in C++, and it was difficult to keep it all up to date. When we produced the system, we didn't make it multi-threaded, partly because of when it was. If we did it again, we'd obviously make everything multi-threaded. And it was often based on research software, which wasn't robust enough and needed a lot of work to make it robust sufficiently. And it was difficult to make a good enough user interface for services. Users wanted something easy to use, powerful, and instant. Well, of course they did, but we couldn't provide all three with the resources we had. Making them easy to use was difficult. Making them instant was even harder. People didn't have the patience for uploading. They didn't have the patience to run services. Maybe, remember, you're running a service maybe on 10, 20 gigabytes of data, so it's going to take some time. And we didn't have appropriate parallelism in there to make it all work in parallel and make it go a bit faster. So we were left at the end with the issues of what way to go. There are really two ways you can take a thing like Carmen forward. You can go with what the clients really want, or you can go with what the technology really allows you to do. And they're often not the same as each other. We looked very hard as we were trying to go forward at the target users that we'd had. There were clinical neurologists and neuroscientists, and for them, they were particularly interested in securing the system, but also in recording data from epilepsy, traumatic injury, Parkinsonism, etc. But they wanted things to be done quickly and they wanted immediate access to it. And then there were neuropharmacologists. They were often working at the opposite end with cultures, assessing the effectiveness of particular pharmaceuticals. And then there was a whole community of research neuroscientists doing research in many different parts of neuroscience, and wanting to analyze and work with the electrophysiological data. And the hope had always been that that data would be supplied to neuromodelers who could use these data sets to constrain their models and test their models out with real data and large volumes of real data in order to make their models better. And lastly, and certainly not leastly, educators, training the next generation of neuroscientists covering the whole range, not just from electrophysiologists, but also the computational neuroscientists interested in trying to make their work more effective and better reflect the reality of the neuroscience. So we were looking at what prospective users want and what they needed. What were they actually trying to solve? And what would they actually use as opposed to saying they might use? We had asked people what they wanted at the beginning of Carmen, but what we discovered fairly quickly about what they wanted, what they said they wanted and what they really wanted weren't entirely the same and often were quite different. And the system had to be made attractive and straightforward enough for neuroscientists to use. It's not that they're not able, it's just that they're busy and they want their work to be done quickly. And what were the issues that discouraged the users? Slowness was one. Non-professional or not particularly wonderful user interfaces was another. They really wanted the user interface to be very good indeed. And then we looked at the technologies that might be useful, noting that neuroscientists most definitely do not want to be using the absolute most modern technology that breaks sometimes. They want that in their scientific systems. They want that in their data acquisition systems, but not necessarily in their storage systems. They wanted to be able to, you need to handle large data sets, ever larger data sets. Electrodes that were 8 by 8. 8 by 8 sets of electrodes were commonplace when we started. Now they're up to 64 by 64 and even bigger. They wanted to be able to do remote visualization of large data sets. That's very important to them. They want to know whether the data they've got is what they expect, what it looks like before they then spend a lot of time and energy analyzing it. They need parallelism at the user level for multiple simultaneous users, of course, but also at the processing level to be able to process multiple data sets or to do parameter searching where you're looking at an analysis tool and running through a set of parameters. You need effective multi-threading for that. And absolutely critically, you need effective search technologies to be able to find the data you want, to find the services you want, to find the workflows that you want. Now, that's Carmen. Carmen didn't take place in a vacuum. There's quite a number of other organizations now who are interested in supporting sharing. And that's moved on, of course, since the time of the Carmen project. Internationally, there's the International Neuroinformatics Coordinating Facility based in Stockholm. Based in the US, but certainly with international wishes, the Neuroscience Information Federation, NIF, and NWB, Neurodata Without Borders. In addition, within the European Union, there was the Human Brain Project, and now eBrains. And there's other interesting projects as well. I haven't time to talk about all of these. CRCNS has been around for quite some time. OpenEphys, plus Open Neuro for MRI and EEG and other kinds of data like that. But let me say a bit about the INCF first of all. INCF was set up in 2006, and the idea initially was that there would be a large number of countries would put money into it, that would allow them to fund major databases and major systems like Carmen, or Carmen on steroids, if you like. It hasn't really worked out that way. At the moment, there's just 3 countries contributing financially: Canada, Norway, and Sweden. Other countries have in the past, most notably the UK, the US, Germany, and there are other countries that are associates and are involved with the INCF, but disappointingly few. It hasn't really taken off in the way that people hoped that it would. They want their country, an organisation that wants to enable open and fair neuroscience, and it still exists and it's still there. A lot of the work they do now is in endorsing community standards and best practices in supporting these. So they currently support NWB, brain imaging data structure, BIDS, and NeuroML, which is a standardized model description language that allows computational neuroscience systems to be shared and to be understood by others. They have quite a number of existing activities. They have a tools and an infrastructure portfolio. They offer very effective training in neuroinformatics. They hold a conference every 2 years, though I'm not sure what's happening about that at the moment. And they have a number of special interest groups: neuroinformatics, reproducibility, neuroimaging, neuroinformatics for aging, neuroshapes. There was also earlier a special interest group, of which I was a member, on data formats for electrophysiological data, and I'll come back to that in just a little while. And then the NIF, the Neuroscience Information Framework, funded by the US National Institute of Health, and they have been cataloguing and surveying neuroscience resources way back since 2006. Originally led by Dan Gardner, who was one of the earliest names in neuroinformatics and was very important early on in getting things going. Now led by Marianne Martone, who's an important person still in this area. And there's a discovery portal allowing you to find neuroscience databases with a catalogue of electronic resources. They have a data sharing service, catalogue of biomedical resources, and ontologies, the ability to search these things and to find things within it. They have ontology engineering, building and enhancing the main terminologies and ontologies. What they don't have is something that allows you anything like an e-neuroscience laboratory. You don't have anything that allows you to run services on their systems. What it is an information framework allowing you to get hold of things and to get hold of tools that do things, but not to actually run them yourself. They've been particularly big in the ontology and vocabulary area, and that has been absolutely critical for inter-lab cooperation. And indeed, we were using their NIF ontology within Carmen itself. You need to have a consistent, flexible terminology that can be used to describe and to retrieve neuroscience relevant resources. That's absolutely critical because people use different names for the same cell types or for different techniques. These are important in finding the right data. And the bigger the volume of the data, the bigger the complexity of the data, the more important is it to organize it in an appropriate fashion. And ontology techniques have been around for quite some time, but it's a major area of work still in neuroscience. And then Neuroscience Without Boundaries, NWB. The overall idea of that is to make databases about the brain more usable and accessible worldwide. Funded by a charitable foundation, Kavli, started a few years ago, and the first things that they're really doing is this Neurodata Without Borders neurophysiology, NWB:N. And that work started off as a special interest group of the INCF. And they've been working with that for some time now, and they're producing some quite interesting results from it. Their overall aims are more ambitious than that. They want to be able to do experimental design, experimental subjects, behavior, new data types, neural activity, all sorts of things. So their overall aims are really high level and really sophisticated. And the idea is that neuroscience data can be shared by all sorts of people. Because even although each brain is different from each other brain, the brain of a particular animal is very similar across that particular animal. So the idea of it being without borders is that it can be shared, and the techniques and the information can be shared all the way across the whole world. They've taken a longer term view of things though. They look at data API. They started off with a data standard schema, thinking right down into how to organize complex collections of neuroscience data. So they're interested in ontologies for that type of thing. Specification languages, which is one particular aspect of it. A standard is not useful unless it is shared and shared in an appropriate fashion and used in the same way by all the people using that standard. So they're interested in specification languages. They're interested in storing large collections of neuroscience data. And they're interested in how people actually get access to it, the applications program interface. How can you interact with it, read it, write it, query it, analyze it? The neurophysiological data standard is the initial target, and they've been working on that now for a number of years, starting off from, as I said, a special interest group from the INCF, holding meetings at the big Society for Neuroscience meeting in the States, and then producing some outputs. And they're now into a kind of second version of it. And they've been holding developer hackathons in order to get services that work properly with these systems, to get the application programming interface really working appropriately. So there's the user view of it. At the bottom level, you've got a data standard specification with particular things related to that type of data. And then you've got the applications programming interfaces in Python and MATLAB. And this looks quite like the NDF system. You've got higher level tools as well. Jupyter widgets, which allows NWBN to be used in conjunction with an electronic lab book. HDMF, conversion tools, extension utilities, and then above that level, visualization and analysis. But the idea is to build in data sharing from the bottom up so that you only have to create the data once. You only have to annotate the data once on your notebook. So it's automatically captured, making sharing the data easier. From the developer's point of view, at the very bottom, you've got specification languages, and based around HDF5, which is an international standard on large volumes, on the storage of large volumes of data of any sort, and then producing a standard specification for that for neuroscience, and then for the data modeling, and then producing the applications programmer interfaces that allow people to access that data. And then above that level, you're back at what amounts to the same as a user view. So a very, again, a very ambitious set of techniques. And in a way, I would say this is almost like what's following on from Carmen. It's been informed by certainly some people from Carmen, and there are people in common between the two. And then eBrains, which grew out of the Human Brain Project. The Human Brain Project was a large 10-year project funded by the European Community. eBrains is really about data sharing, all sorts of data, behavioral, computational models, electrophysiological data, connectomics data, omics data, functional data, reconstructions, brain atlases, etc. It includes sharing brain simulation platforms, the virtual brain and also Nest, and indeed goes right down to the hardware of brain-inspired computing for neuromorphic computing. But the idea here is to allow tools and services that can be used to address internationally challenges in brain research. So where are we now? The critical thing is enabling replicatability, repeatability, and reusability in neuroscience. If you can't repeat an experiment, is it science? But of course, you can never repeat precisely the same experiment in any biological area. But what you want to do is to be able to repeat experiments on different animals or with different neurons. You want to be able to repeat the experimental data analysis to see if new techniques of analysis give you the same results. You want to be able to do validation across a whole lot of different experiments in order to bring together data from different animals perhaps, or from different neural cultures, or from different brain slices, to bring them together and see if you can get appropriate results that go across that whole lot, enabling you to think that you've got something which you are more certain about. Are we further forward? Yes, The Neuroscience Without Borders designs are better, they're more detailed, they're easier to use, and they're publicly available. That's an aim there. One of the mistakes that I think we made in Carmen was not making everything open source to start with. If we were to do it again a bit later, we would certainly have done so. And indeed, communications technology has moved on as well. Tools are faster, tools are better, they're easier to build, they work better, they don't break as often. But at the same time, the data volumes have increased. 409 6-element electrode arrays, 3D resolution imaging using two photon techniques are generating really very large volumes of data. It's important to have analysis tool transparency and sharing. People want to use open software. They want to know exactly what that analysis tool is doing so that they can compare it with another analysis tool, particularly if the results are different between different analysis tools. More use of repositories. Open repositories help produce better quality software because people can look inside it, correct it, extend it. Open Neuro allows that kind of thing. You want to be able to share the models that you're working with, and that's important in computational neuroscience. The virtual brain and NEST have done a lot of work in that area, and INCF has attempted to as well. Reusability of data is critical. NWB:/M really does help, but we're not quite there yet. And because no two neurons and no two brains are the same, we need to be able to reuse data sets from different experiments to explore what stays the same and what's different. But organizations that bring together scientists remain fragmented. There are problems within that whole organizational area. Some of these are political problems, but they can be solved. And really, if we want to be serious about properly sharing data without borders across the world, the science community needs to get together and be together and support things like INCF, or at the very least, NWB. Okay, many acknowledgements here. All the Carmen researchers, it was a big project, there were more researchers than this. And the INCF data sharing task force members, some of whom then went on to be very involved in NWB. Now, so to conclude, I've spoken a lot about the Carmen project, I've spoken a lot about what it did, and I've tried to give you a feel for what's going on in this overall area of neuroinformatics. Thank you very much for listening to me.